In this video, we'll be talking about different kinds of membrane proteins. So far in this chapter, we've been focused on enzymes, which are proteins that catalyze reactions. We'll now move on to talking about other types of proteins, specifically ones that are associated with membranes, because these are also common drug targets. Membranes are made up of phospholipid bilayers. These phospholipids have a hydrophilic head and hydrophobic tails. The structure of these molecules causes them to spontaneously assemble into bilayers. So here's the first layer, and here is the second layer. And in this way, the hydrophobic parts of the molecule are all together and hidden from the aqueous environment, both outside of the cell and inside of the cell. Membranes define the boundaries of individual cells, as well as organelles inside cells. And they're impermeable to many molecules, which is important because it allows the body and the cell to compartmentalize functions. For example, the cell needs to be able to break down toxins and waste products, which is done in the lysosomes. If the mechanism involved in breaking down waste wasn't compartmentalized to the lysosome, the cell would just start breaking down everything, including the things that it needs. Having those mechanisms be membrane-bound means that only things that get into the membrane-bound lysosome are going to be degraded. Now, of course, we need to be able to get nutrients and other important molecules into our cells, and we need to be able to transmit things out of the cells as well. Membrane proteins allow for the regulated passage of molecules and signals through the membrane so that cells can maintain their identities and unique functions while still getting the nutrients and communication signals they need to function. There are different types of membrane proteins, and they're classified by their function. The first types are transporters and channels. These membrane proteins are responsible for transporting molecules across the membrane. The main difference is that transporters require energy in order to transport molecules against their concentration gradient, while channels transport molecules down their concentration gradient without using energy. The concentration gradient refers to the fact that many substances exist at unequal concentrations inside and outside of the cell. So if we consider this line to be the cell membrane, and above it is going to be outside the cell, and below it will be inside the cell, some substances, for example potassium, are going to exist at a higher concentration inside the cell than outside the cell, while other substances such as sodium ions are going to exist at a higher concentration outside the cell than inside the cell. Many processes in the cell rely on the presence of a concentration gradient, including ATP synthesis and the generation of neuronal and cardiac action potentials. There's also certain molecules the cell wants a lot of, such as glucose and other nutrients, and other molecules the cell doesn't want as many of. We'll focus first on transporters, which are membrane proteins that help create and maintain these concentration gradients by actively transporting molecules against their concentration gradient. That means they move molecules from a compartment where they are at a low concentration to a compartment where they are at a high concentration. This process requires energy. There are two types of active transport that are defined by where the energy for the transport comes from. Primary active transport gets energy directly from ATP. An example of a primary active transporter is the sodium-potassium pump, which is responsible for maintaining the membrane potential of a cell. Like I mentioned before, sodium exists at a higher concentration outside the cell than inside the cell. Potassium is at a higher concentration inside the cell. The sodium-potassium pump actively pumps 3 sodium ions against its concentration gradient from inside the cell to outside the cell. It also pumps 2 potassium ions from outside the cell, where it is at a low concentration, to inside the cell, where it is at a high concentration. And the energy for this process comes directly from ATP. There's another kind of active transport called secondary active transport, which gets its energy from the potential energy stored in a concentration gradient established by primary transport. For example, the sodium-glucose cotransporter takes advantage of the fact that there's been a sodium concentration gradient set up in order to transport glucose against its concentration gradient. This cotransporter takes these sodium ions that are at a high concentration outside the cell and moves them down their concentration gradient to inside the cell where they are at a low concentration. So this process is energetically favorable. But at the same time that it's doing that, it's taking this glucose molecule and transporting it against its concentration gradient, so away from outside the cell where it's at a low concentration and towards into the cell where it is already at a higher concentration. This process isn't directly using ATP, but it wouldn't be possible if the sodium gradient wasn't set up by the sodium-potassium pump using ATP. So it's still considered a form of active transport. In general, anytime you have a molecule that's being transported against its concentration gradient, that's always going to require energy coming from somewhere, and it's going to be a form of active transport. These two examples also highlight that some transporters are co-transporters, or otherwise known as symporters, and transport molecules in the same direction. So for example, the sodium glucose co-transporter is going to be a symporter because both the sodium and the glucose are moving in the same direction. Other transporters, like the sodium-potassium pump, are going to move ions or molecules in opposite directions. So here, the sodium-potassium pump is moving sodium out of the cell and potassium into the cell. And these kind of transporters are called antiporters. Moving on now to channels. Channels are a type of membrane protein involved in passive transport. So they move substances down their concentration gradient. This process is already energetically favorable, so it can occur without using any energy. The tertiary structure of channel proteins is such that they may only allow certain kinds of molecules or ions to pass through. For example, if the passageway of a channel has a lot of negatively charged amino acids, it's going to attract cations while repelling anions. Other kinds of channels are sized exactly for a certain kind of ion or molecule to pass through, but not others. An example of a channel is an aquaporin, which are channels that are specifically shaped so that only water molecules can pass through. And notice that these molecules are moving down their concentration gradient. So the water in this example is at a high concentration outside the cell and at a low concentration inside the cell, so the water molecules are moving down their concentration gradient. Because channels only do passive transport, this channel cannot move water molecules in the opposite direction. As an additional layer of regulation, some channels can be gated, which means that they can be either open or closed by the cell. For example, there are channels that are called voltage-gated channels that are only open once the cell hits a certain membrane potential, and channels called ligand-gated channels that are only open when a specific molecule is bound to the channel. Next, we'll discuss receptors. Receptors are responsible for receiving a signal from outside of the cell and transmitting it into a response inside of the cell. An example of a receptor is the G-protein coupled receptor, which we see pictured here. And this G-protein coupled receptor is going to receive a signaling molecule from outside of the cell, and that's going to activate this G-protein signaling complex to induce downstream signaling effects. Receptors are really important for communication between cells, and they're a very common drug target. We'll talk about them in more detail in the next video. There's also a class of membrane proteins that actually are also enzymes, and they're involved in catalyzing reactions that are localized to the membrane. An example of a membrane-localized enzyme is adenylyl cyclase, which converts ATP into the important signaling molecule cyclic AMP. The reason it's important for this enzyme to be at the membrane is it's actually coupled to these G-protein receptors. So when this G-protein complex is activated, it allows them to quickly activate a nearby adenylyl cyclase enzyme and induce signaling. The last kind of membrane protein we'll talk about in this video are anchoring proteins, which help the cell maintain its structure and also help nearby cells form connections with one another. For example, cadherins are a protein that are involved in making junctions between cells. So to sum up, the barriers formed by membranes are an important part of establishing cell and organelle function and identity. And membrane proteins help pass molecules and signals from cell to cell and within a cell in a regulated way. Transporters use energy to move molecules against their concentration gradient, with the energy for primary active transport coming from ATP, and the energy for secondary active transport coming from the potential energy of other molecules moving down their concentration gradient. Channels move specific molecules down their concentration gradient without using any energy. Receptors don't necessarily move molecules across a membrane, but they do transmit a signal from outside of the cell to inside the cell. There are also membrane-associated enzymes and membrane anchoring proteins responsible for maintaining cell structure. Because membrane proteins are so deeply involved in regulating a cell's function and identity, all classes of membrane proteins are useful and important drug targets. One thing that is important to note is these membrane protein types are not mutually exclusive, so a single protein could fall into multiple categories.